So, good day. We'll be talking about the complement system. And, so, the complement system, basically, what they do is that they complement the function of the immune system, of your immune system. They complement it. And also we can say they augment the function of the immune system. So this complement system, we have like three basic type. We have three basic types. We have we have three basic pathways to activate this complement system. We have the classical pathway. We have the alternative pathway. Then the mannose binding lectin pathway. So these are the basic pathways. These are the three basic pathways that we have for this complement system. So I'm going to start with the classical pathway, then alternative pathway, and the mannose binding lectin pathway. So before I start, these complement systems are made up of what? Complement protein. So what are complement protein? Complement protein are just proteins that are produced by what? They are produced by the liver, and not just the liver. They can be produced by the what? The macrophage and even the epithelium of the intestine. And so we'll be looking at we have different complement protein basically. So as I explained, I'm going to explain this classical pathway first, then you understand how complement protein works in activating this pathway. So let's start with the classical pathway. So, as I said earlier, the complement system can augment the function of the immune system. So now let's assume we have a bacteria. Let's assume this is a bacteria. This is a bacteria. And as I explained earlier in our previous class, I told you guys about antibody. And basically, the antibody that we'll be looking at here in this complement system, the antibody we'll be looking at is your IgM and IgG. Basically, that's the two antibody that activates the complement system. IgG, said IgG or IgM. And you already know the structure of your antibody. These are the structure of the antibodies. Basically, like Y. Let me just say Y. But basically, let me just make it like that. But basically, you know your antibody look exactly like this. Yeah, that's the way your antibody looks like. Which are tetrapeptide. See, this is the light chain, and this is the airy chain. So we say they are what? Tetrapeptide. Yeah, they are tetrapeptide. And you know they are joined together by what? Disulfide bond. So let's first say that that's not the topic for today. Just say let me talk about it. Now let's continue our classical pathway. This is either IgG or IgM. Now this portion, as I explained to you in the previous class, this part is called the FC. This is the FC region. This still is the FC region. So this is where your complement protein is going to bind. And as I said earlier, complement protein are produced in the liver, they're produced by macrophage, and the epithelium of your intestine. So now we have complement protein, we have C1, we have C1. So this C1 here, this C1 is going to be the one that will bind to this what? To this FC portion. But this C1, one thing about this C1 is that the C1 has like 3 domains. It has the Q, R, S. So is the Q domain of the C1 that will bind to the FC region. So these are resume C1. And anytime you see, maybe you're reading this topic in the textbook, anytime you see a bar on top of any complement protein, it's telling you it's active, like the protein is active, it's in its active form. So let's continue. Your C1 is going to bind to the FC portion. Now what part of the C1? Your what's the Q domain of the C1 is going to bind in. So now this C1 is going to activate another complement protein, which is what? Your C4. Now, the C4 is going to be coming from the blood, if you want. So your C4 is going to act on what? We have C4. So this C4 is having what? The A part and the what? The P part, which is simple. This is what I'm trying to say. This C4 is having a component which is what? We have the what? The C4A and we have the what? The C4B. So let's say this is what is equal to C4. So now once this C1 act on C4, this part is going to be cleaved off. This C4A is going to be cleaved off. Then we now have the what? The C4B. So which is what? C1, then we now have what? So now the what? Okay. C4B. So let me use this color for the A part. So the C4A. This is the C4A that is living. We actually don't need it for now. Then this is the CB part. The C4B part. Now, this C4B is going to activate another protein, which is what? Your C2. And same principle, C2 is also going to have what? It's going to have C2A and what? C2B. So basically, the C4B will allow us the cleavage of this C2A to move. Then the C2B is going to come to this part. So we said C2B is going to leave. C2B is going to leave. So we're going to have our C2A. Now, if you notice, we will not talk about C3. From C1, we have to jump towards the C4, then the C2. So meaning what? Let's talk about our C3. So basically, your C3, this C4 and this C4B and C2. Oh, sorry, this is supposed to be C2B. So this C4B and C2B are going to activate what? They are going to activate. It will act on what? The C3. And you already know C3 is going to have 2 parts, which is the what? C3A and what? C3B. C3A and C3B. So basically, this one is going to, the same thing is going to happen. This guy is going to be cleaved off. This guy is going to be cleaved off. Then this guy, this C3B is going to come here. C3B is going to come here. So we have what? C3B. So we call these two, this C4B and C2B, we call call them the what? The C3 convertase. So the C4b, the C2b is called the what? The C3 convertase, because they are the one that will act on the C3, so to be able to cleave into what? C3a and C3b. So let's continue. So we've talked about 1, 4, 2, 3. So the next one is definitely going to be the what? The fifth one, the C5. So this guy again, they will act on what? The C5. The C5 is going to be cleaving to what? C5a and what? C5b. So the C5a is also going to be what? C5a is also going to be what? It's going to be cleaved out. Then C5b join this complex. So it makes sense for us to say C4, C2, C3 are what? Are called C5 convertase. Convertase. So these three guys are called what? C5 convertase. Now, this is what I have to clean this place up to make this. C5 convertase. Now, this C5b is going to activate C6, activate C6, C6, C7, C8, C9. So, okay. 5b, once 5b is activated, C6, C7, C8, C9 are going to be activated. So these guys will create what we know as what? They will create this guy, they are going to create what we know as what? Membrane. They will create a tunnel here, which is known as what? The membrane attack complex. So they are going to create this. The main purpose of this complement system is to create this membrane attack complex. And it's this membrane attack complex that is going to do what? The membrane attack complex is going to like work against this gram-negative bacteria. So the deficiency, if there's any deficiency of any of this protein, we won't be able to like create our membrane attack complex. So that's about that. Okay, there's a clinical correlate. Yeah. Okay. As I said earlier, if there is a deficiency of any of these complement protein, we won't have a membrane attack complex, which is going to this membrane attack complex work against gram-negative bacteria. So an example is okay, let me use people with chronic gonorrhea infection, which is just like repeatedly gonorrhea infection. This kind of people, most times the issue is not so now we'll continue the complement system by explaining the alternative pathway. This alternative pathway is antibody independent. It does not depend on antibody. So it acts as what? First line of defense. So we can say the alternative pathway it's just like your innate immunity, basically, because it does not need antibody for it to work. So this is what happened. Let's assume this is our bacteria. So the bacteria releases what we know as the what? Endotoxin. So is the bacteria will release what we know as the endotoxin. So is this endotoxin that will activate this organic pathway, unlike our classical pathway, which is activated by what? Your IgG or IgM. So this is what will happen. Your basically your C3b in the blood, like they hydrolyze, like this to hydrolyze easily, hydrolyzes easily. Then so since C3b C3 hydrolyzes easily, then we have what? C3a and C3b. So our C3b is just what we need, as you know. And I don't know if you've been thinking about this. What's the function of this C3b? In the classic pattern, I was saying C3a, C2a, C4a, and C5a. Basically, what this A portion, what they do is just that they help in inflammation. They don't help in inflammation. This is three parts. Helps in inflammation. So now, your C3b is already here. C3b is here. C3b will activate another complement protein. This one is somehow different, which is what? CB. And as you know, it activates CB, which means what? CB is going to cleave into the two parts, which you all know that is going to be what? CBA and what? CBB. So what we need is the what? The CBB. Sorry, supposed to use black for this. So CBB. Now this C3b and this CBB also have the capability of what? Hydrolyzing C3. They can also hydrolyze what with C3 into what? C3b and what? C3a. So it's somehow fun, you just need to know it. This C3b and CBB can also hydrolyze what? C3 into what? C3a and C3b. So which means this place we are going to have what here again? Your C3b. So it makes sense. It makes sense for us to say these two guys are what? Your C3 convertase. So these two guys are your what? Your C3 convertase. That's about it. So in your MCQ, you can either see C3b, CBB as okay. This is what you might have this kind of stuff. Okay, let me just. I don't want to make it too complex. If you remember, in our classical pathway, we said what? The classical pathway, we said the C3 convertase is the what? C4b and C2b. Now, this alternative pathway, our C3 convertase is just what? C3b and CBB. So if you see any of this in your MCQ, then just pick any one. Is either you see this or the other one in your MCQ. And yes, let me say previously in classical pathway, let me make this clear to you. In classical pathway, C3b and C2A are called what? The C3 convertase. But now recently it is now what? C3B and C2B, which I explained earlier. So now let's continue. Now, with your knowledge of the classical pathway, you should be able to finish this up without me explaining. So it's just going to be like what? C. It's just going to be C5B. You already know the C3A is being cleaved off, right? You already know the C5. Okay, okay, I don't want to be fast. Let me be fast. The C3B will act on what? The C5, which will later cleave the A part. So you have the C5A going up to this dimension. Then the C5B coming to this part. The C5B is C5B coming to this part. C5B. C5B. Then what's going to be activated again? Remember from your classical pathway, you just make a continuation from the classical pathway. What's going to be activated? C6, yes, you are getting it. C6 and C7, C8, yes, and what? C9. Which eventually creates what? The membrane attack complex. The same is just similar to your classical pathway. The difference is just this initial part. Yeah. Then let's move to the last pathway. Which is what? Mannan binding lectin pathway or mannose binding lectin. So basically, let's continue and we're talking about the last one, which is what? Your Mannan binding lectin pathway. So on the surface, you can also call it the mannose binding lectin, because on the surface of your bacteria, you have what? Mannose, which is carbohydrates. Right on the surface of our bacteria. So now we assume we have a protein that is synthesized by what? Liver, which is what? Mannose binding lectin. So this mannose binding lectin is called mannose binding lectin because of what? It's going to bind on the mannose. So we said mannose binding lectin binds. Let's say these are mannose. Use the mannose. So since it's binding on the mannose, we call it mannose binding lectin. It's a protein that binds on the mannose. So this mannose binding lectin protein is going to hydrolyze C4 to give us what? C4A and what? C4B. So now you already know the one that the C4B is what we need here. So this is C4B. This C4B will activate what? C2 into what? C2A and C2B. The C2A move up, C2A. Then the C4, the C2 is going to be. These two guys are going to act as what? C3 convertase, just like the classical pathway. C3 convertase. And just like the classical pathway, to make it fast. Just like the classical pathway, then you have what? C3B, C3A, then what? C5B, C5A, then what? C6, C7, C8, then your C9, which we want create all known as the what? Membrane attack complex. So that's all. I'm going to make another video about the clinical correlate to this. So thank you.